So the goal of this lecture is to present statistical methods to analyze a large amount of single particle trajectories, and those trajectories are usually extracted from data, and here I'll focus specifically on super resolution. So what is the framework, statistical framework under which it is possible to analyze the data? So let me first introduce the data. So the data are actually a collection of small amount locally of trajectory. Actually, that can be also, if you wait sufficiently long enough, a large amount of trajectories. So it's like an ensemble of points that are connected in time, and we suppose that this is done by a tracking algorithm. So you have a time t1, time t2, time t3, and so on. And usually we assume that the time step delta t is constant between tk plus 1 and tk. And so we get an ensemble of short trajectory, about 7 consecutive points. And here this is an example where this delta t was about 50 milliseconds. So when you superimpose all of this trajectory, you can get this classical representation where you have a dendrite, and this is here a dendritic spine, that contains many of those trajectories. Now, what can be said with this trajectory? Not just like using statistics to compare to different biological conditions, but can we use this trajectory to infer something about the biophysical property of the membrane? So this is another example where you have again this beautiful ensemble of trajectory. The color code is that each color corresponds to a trajectory, and as we have published a couple of years ago, if you superimpose the region where you have all of this trajectory, it exactly superimposes where the synapses, and this is given here by the white spot. It's a marker of what is called a postsynaptic density, which is an interesting region on a neuron. But this is a general framework that you acquire trajectories on the surface of a cell, and you would like to get information about the heterogeneity of what happened on the surface based on the analysis of these trajectories. So for this, the first thing that is needed is a model for single particle trajectories. And the classical model is the Langevin equation, which states that the acceleration plus the damping term plus forces, this is the example of conservative forces, equals the Brownian term, which is due to the collision. So you have a particle embedded into a fluid, you have all the particles that are colliding with these particles. It's usually isotropic, and this ensemble of condition is the framework for the classical fluctuation dissipation theorem, which states that there is a specific connection here between the friction and the diffusion coefficient. And this assumes steady state, the temperature is fixed, and an homogeneous medium, which we are going to see in a second that this is not satisfied on the surface of a membrane. But from the Langevin equation, in the Smoluchowski limit that is in the large friction, it is possible to get rid of the inertia term, and we are left with a classical stochastic equation where the velocity is driven by the force plus the noise term. So what is the problem here? Why the classical Langevin equation in the framework of fluctuation dissipation cannot be applied? So let's look at the following example. Suppose here we place obstacles, and in black this is a real trajectory that would be generated by physical forces. So the trajectory avoid the obstacles. However, suppose that we acquire trajectory only at a certain time step delta t, which corresponds to acquire the trajectory at the stars. So we have then an ensemble of points, and what the tracking algorithm is doing is to connect those points with the dotted black line. So clearly the trajectory in dotted line is very different from the real trajectory. This is the real physical trajectory. It's very different. One, the first reason for this is the sampling. And of course, this, when you get this trajectory, you do not see the underlying obstacles that has created the change compared to a situation where there were no obstacles. And second, the discretization process allow you to get only a certain sample of the trajectory at a certain resolution delta t, which is the sampling resolution. So the dotted line trajectory is really what is called a coarse-grain trajectory. And this is exactly what the microscopy is giving us, not the real trajectory. So for this, we need a generalized stochastic equation to describe this. And this is exactly what this equation is. That is, we take the exact form of the Langevin, the stochastic limit of the Langevin equation. That is, the velocity is given by a first order term which is deterministic, which can be a drift or a potential well. And the second is due to a stochastic term, and usually we adopt the Brownian motion. So now the question, once we have decided that the model for our physical process is this coarse-grain equation with no necessarily any specific relation between the drift term and the diffusion coefficient, then in that case what we're interested in is to recover B and D from the trajectories. So this actually is possible, but before that I would like to attract your attention on another point that has to be solved, which is the additional noise that is usually added by the instrument or any acquisition system on the trajectory. So suppose the observed trajectory is in blue. So you get here your trajectory, that is the points at different time step, T1, T2, and so on, T3. But due to the instrument, we assume that there is a Gaussian noise that is added to each point, so that instead of detecting the point in blue, we detect the point in green. And instead of getting this point here, the acquisition system take the other green point, and so on. So instead of having this trajectory, which is the physical one, we get that trajectory in green. So again, now the question is, how is it possible to deconvolve, that is, to get information about the underlying stochastic process, for example, the first moments, and remove the effect of the noise? So for this, we need a model for this first question about deconvolution. So suppose xn is the position of a particle, yn is the observed one, and they are linked by the following equation, that is, at each time step delta t, yn is equal to xn plus sigma, the amplitude of the noise, and eta is a Gaussian noise, classical with variance 1. So then, at different time step, 0 delta t up to n time delta t, if we have a coarse-grained model, the position at time t plus delta t, here n plus 1, is equal to position at time n plus drift plus the noise. And so one possibility to make this deconvolution is just to write that the observed point is the real point plus the noise, and to calculate the transition probability to switch from a point x at time tn to point y at time tn plus 1. And this is exactly the probability that xn plus zn, actually plus 1 equals y, condition that xn plus zn equals x. And it's a classical exercise to see that this is actually the convolution of two Gaussian integral. And after calculating one integral, we are left with this equation. And this assumes that actually the probability density function for the process xn plus 1 xn is a Gaussian, is exponential of x of y minus x minus a of x delta t divided by 4d delta t divided in dimension 1 by square root of 4 pi d delta t. So this assumes this, which is only true in the entire space R. Otherwise this is incorrect. For example, if you are in an interval, you have to account for the boundary conditions. But then, starting from this, you can see here, this is the integral for the one Gaussian that is left, and this accounts for already the effect of the noise with the physical trajectory. So after a calculation, assuming that sigma is small, and the calculation will be soon be available in our paper with Nathanael Ose, we can derive an expression for the apparent variance, which depends on the variance of the noise, the derivative here of the drift at position x, and the diffusion coefficient, assuming that here b is a constant. It's a constant d, square root of 2d. Then you get here the contribution of each terms in the variance of the convolved process, the process of observing the real trajectory xn when a noise is added. in higher dimension, the formula is a bit more involved, and in confined space, this is harder to calculate because of, for example, the effect of the boundary. But now it is possible to use this expression for the probability transition function to calculate the first moment. We'll see that now. That is, suppose you compute empirically the difference yn plus 1 minus yn divided by delta t when yn is conditioned at position x. That is, you fix position x and you look, if you discretize, for example, a beam around x, and you look at all trajectory such that the point yn fall into this beam, and then you calculate, you see where is yn plus 1, and you calculate this difference. What the formula says, and this can be shown here, is that this calculation, which extracts for you the drift, is such that you get exactly the drift of your coarse grain motion up to order sigma. That is, the noise here is eliminated in the process of averaging, so that you are sure that when you calculate at resolution delta t your drift, you get your coarse grain drift, which is an approximation of your physical drift. And similarly, if you calculate here the second moment, which gives you the diffusion coefficient at point x, it is connected to the noise, the real drift, and here there's an additional term which corresponds to the derivative of the drift at position x. And in higher dimension, you get here the divergence. So this is very important to come back to this result, because this says that if you see an ensemble of arrows, this is the arrows that you get from your drift, and there is no contribution of the isotropic noise that is added. So then it's we can take what we have done here to estimate the drift, which is given by the following formula. This is the limit as delta t goes to 0 of the expectation of the difference of xt plus delta t minus xt divided by delta t. So for a classical stochastic process, which is x dot equals b of x plus 2 square root of 2 dw dot, you can recover here b of x from your trajectory by the following formula. And actually, taking the formula that we have just seen, we can build with not much effort the following empirical estimator by averaging over all trajectory that are such that are going through the point x. And this empirical sum is a good approximation of the drift at position x. So in this way, we can invert the stochastic equation using the massive amount of trajectories. And I would like to note here that this analysis can be extended to polymer physics, where you get, in general, anomalous diffusion. So this will be the subject of a different lecture. So similarly, using the second moment, it is possible to get access to the diffusion tensor by this formula, and this is the associated empirical estimator, where we sum over all trajectory going through the point x. And now we can recover the entire stochastic process, which is the drift and the stochastic part. And once this dynamic is reconstructed, we get a lot of information. And for example, this will be the subject of a different lecture to explain how we can use this to generate simulations on a structure, on the image of a cell, for example. So typically, from the calculation of the diffusion tensor, we can get a map, and this is here the color code, where you can at each point give a number for the amplitude of the diffusion tensor. It is also possible to get more information from the diffusion coefficient, because if the diffusion coefficient or diffusion tensor vary at a point d of x, the reason for this is due to the heterogeneity of the membrane organization. And the first model can be an ensemble of obstacles located on a square grid characterized by two parameters A and L. And now, for example, Brownian simulation at a sufficiently long scale show that the MSD is linear in time. So you recover from here some homogeneity at a certain scale. And the associated diffusion equation is the classical 2D diffusion, and it is possible to relate the apparent diffusion coefficient, which really would be the diffusion coefficient from a particle starting, for a particle starting in a block here, to move to another block. So we have the ensemble, each square is really coarse-grained into a single element. So then it is possible to relate the apparent diffusion coefficient df to the real diffusion coefficient through the geometry. And this calculation can be found in this reference, where basically the apparent diffusion coefficient depends on the mean time for a particle that starts here to escape in this domain. And the escape time depends on the geometrical properties such as the length and A. And this is the exact graph, how the apparent diffusion coefficient depends on the ratio A divided by L. And you see here this very nonlinearity that can be seen in this region that is decomposed in the logarithmic part and algebraic part. And the algebraic part is of course given by the square root here of L minus 2A. So these considerations have been applied to estimate, estimate the fraction of obstacles once the diffusion coefficient or the mean diffusion coefficient has been extracted for a dendritic spine and for a dendrite. And we could find that this leads to 70% in one case of membrane occupied compared to 50% in the other case. So then the next question is, once you have an ensemble of drift calculated by your empirical estimator, what can we say about region where you have arrows that goes in all directions? So for this, again, now we need a model for the organization of the vector field. What does it mean? So what does it mean? So it can be due to local transport or a field of force. And then in that case, it is possible that the force is deriving, it's coming from a potential well. The potential well can be to electrostatic interaction, although this would mean very short range, or mechanical interaction for longer range. But we should say this, in general, there is no energy landscape, because the Boltzmann distribution is not necessarily a solution of the Fokker-Planck equation, and this is especially true when the diffusion is not constant. Then we cannot have exponential minus phi divided by d, which is solution of the Fokker-Planck equation. So there is no simple connection between the density and the potential well. So no relation, or no clear relation, no physical relation between the potential phi and the density, because this is in general not equal to the density. So at least locally we can expect that we could find this relation when the force is coming from a potential and the diffusion is almost constant. So for this, using a classical representation of a potential well, which is approximated by a parabola and in dimension 2 by a paraboloid, using differentiated v and using the gradient, we get this converging arrow which is a signature of a potential well. And this is a feature that can be found or can be looked at to see if it appears in the data, in the ensemble of arrows that would come from the procedure of extracting the first moment from the trajectories. So this is what we've been doing in the last couple of years, decomposing your cell or the surface of a cell in bins or any kind of way to discretize your space. You can use different sizes, and what we could find is that at different scale there are converging arrows, although the amplitude is not the same. What is important is that you recover this convergence, and this is a signature of having here a potential well, which is again it might be a coarse-grained potential well, but this is a signature that something here is interacting with something else. This is a signature of interaction, although the nature of the interaction is obviously not clear. So we could apply this on the super resolution data acquired on piraceptors, and we could see that the region here of high densities are exactly matching region where potential well can be detected. And this is example here of 3 potential wells. Again, suggesting that the region of high density are not due to obstacles, but due to physical forces of large extension size of hundreds of micrometers, hundreds of nanometers, and it's not clear what can generate those long-range forces, because electrostatic forces are too short to generate forces with such a long distances. So, to conclude this introduction for the literature about the analysis of single particle trajectories, the massive super resolution data allows to resolve or to solve this reverse engineering problem and to extract this local property by calculating basically 2 moments. The first moment, which is basically the drift or the velocity, and the second moment, which is the stochasticity and the diffusion coefficient. For this, what is quite remarkable is we do not need any a priori knowledge of the probability distribution function of the physical process or the coarse-grained process. So in particular, the probability distribution function is not necessarily Gaussian, and it's unimportant that it is Gaussian or not, because the calculation of the moment does not require to know this property. And any estimator using a priori Gaussian and several steps will necessarily introduce a bias in the analysis that, for example, can be reflected in the estimation of the energy, for example, in the potential well. There is another problem that we haven't discussed here, that usually the data are projected on the focal plane, although they are coming from the surface of a neuron. So there is usually another step which is needed to remove the drift that is added due to the projection of a stochastic process that happen on the surface of a membrane to the plane. So if you have, let's say, a ball and you have a stochastic process occurring here, and this has to be projected, usually there is an additional component or a drift here that is coming due to the projection. And this is will be the subject of a different lecture. So to finish, I would like to say that the origin of the potential, well, to this day remains a mystery. The potential have been seen in different ensemble of trajectories, such as empirireceptors, calcium voltage channel on the presynaptic terminal of neurons. We have also observed this for the GAG protein, which are important for the capsid assembly of the HIV virus. So this potential seems to be a general feature of the heterogeneity occurring on the surface of cells, and we still do not know where it comes from. So I'll conclude this lecture by saying that what I've discussed with you today is an example, a method that we have developed to extract single particle trajectory features. It is based on this really computing two moments, and I hope that I have convinced you that this is the right way to analyze your experiments. Thanks.